Chemists, physicists, biologists, and computer scientists from around the world have now completed the most ambitious biological research project of all time, the Human Genome Project. One of the lead scientists who worked overtime on this project was Dr. Lloyd Smith, a chemist at the University of Wisconsin in Madison. We asked him just what is the Human Genome Project.
Yeah, well, the pithiest sort of summary of it is that it's a project to determine the sequence, so the structure of all of the DNA of the human species. And that ends up being a pretty good sized task, because it turns out that every cell of your body has about 3 billion bases of DNA in it. And the total amount of DNA sequence that's been determined in the history of mankind, which is short in this respect, about 25 years, is maybe of the order of 100 million bases.
That was 5 years ago. Thanks to the invention and hard work of Dr. Smith and a few hundred other genome scientists around the world, the pace of progress increased dramatically from 1998 to 2001. In 2001, in a White House ceremony, Dr. Francis Collins, head of the Human Genome Project, and Dr. J. Craig Venter, president of a private company, Celera Genomics, joined with then-President Clinton to announce the first working draft of the human genome. And then in April of 2003, the final draft of the human genome was announced and published. In effect, the completion of this monumental project. We now have, in other words, nature's blueprint for constructing a human being. And here it is, a 4-letter code written in very long, unpunctuated sentences on 23 pair of human chromosomes. This blueprint for making a human being took nature 3.5 billion years to create. Dr. Smith, along with hundreds of colleagues in laboratories around the world, took 13 years to learn to read and record the entire code. But now comes the hard part, understanding what it says and how it works. To understand what has been accomplished already, as well as what remains to be done, we need to go back and start with the biggest picture of all, as Dr. Smith implied, the history of mankind. He, however, was talking only about the strides taken in the last 25 years in molecular biology laboratories. In a sense, the history of the last 4 billion years provides a broader insight into the importance of the Human Genome Project. Consider, 4 or 5 billion years ago, the dust from an exploded star gathered together to make a small planet we now call Earth. A billion or so years later, in the ancient oceans, some of the most common of the atoms that made up that dust, carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, a few more, gathered together to make structures that we call living. The important thing to note for our story is that in those first days of life on Earth, the basic patterns were set, and all life thereafter, including human life today, is built on those first basic templates. And the most basic template of all looks like this: a very large molecule called deoxyribonucleic acid, DNA for short. It is the DNA in the nucleus of all living cells that directs the life activities of all living cells, and hence of all living organisms, plant, animal, and human. All means not only the organisms living today, but all the plants and all the animals that have ever lived in the 3 to 4 billion year history of life on Earth. You see, DNA is like the software code in a computer, the program that tells the computer hardware what to do and how to do it. Humans invented the computer code. Nature invented the code on DNA that directs all living activities. And the Human Genome Project is deciphering that code. You can see why Lloyd Smith calls it a pretty good-sized task. Before looking at some details of just how scientists are deciphering that code, let's review what we know today about how DNA is built and how it works. First, the structure. Scientists discovered a few decades ago that DNA is a very long molecule, shaped like a twisted ladder. The rails of the ladder are made of alternating chemical groups of sugar and phosphate. The all-important code is carried by the DNA molecule on the rungs of this ladder, as in this model diagram. And there are 4 different kinds of rungs. Each rung has a particular chemical structure. A, G, C, T. Adenine, guanine, cytosine, and thymine. Chemically, these are 4 examples of well-known structures called bases, the opposite of acids. Well, just as the 26 letters of the English alphabet can be put together in an almost infinite variety of sequences to make an almost infinite number of words, so too the 4 letters of the DNA alphabet, the 4 bases, A, G, C, and T, can be placed in lengthy sequences in an almost infinite variety of ways to make an almost infinite number of messages. In practice, there are only a few 100,000 English words in even the largest dictionary. In nature, there are tens of millions of species of plants and animals that live or have lived on planet Earth. But the amazing thing is that all of these millions of separate species of plants and animals use very similar codes on their very similar DNA molecules to stay alive and to reproduce. Now, the DNA in microscopic yeast cells, for instance, has already been sequenced, and it's shown to have many of the same codes as the DNA in humans. All of the DNA in a common microscopic roundworm has now been sequenced and shown to have many of the same codes as the DNA in humans. In fact, we share over 90% of our DNA code with mice and rats, and over 99% with our close relatives, the chimpanzees. Well, how does it work? How does the master DNA code get translated into living activities, whether the living organism is a yeast, a worm, a mouse, a chimpanzee, or a human? The code works at the basic level of life, the cell. The human body is made of 100 trillion cells. Each of these cells has a nucleus. Inside that nucleus are spindly structures called chromosomes. Now, chromosomes come in pairs. Humans have 23 pairs. 1 of each pair came from the mother and 1 from the father. A single chromosome is, in fact, a single, very long DNA molecule coiled up inside a protective protein coat. If you take out the DNA from a single chromosome, uncoil it, and stretch it out, it would be a few centimeters in length. If you took all the DNA from a single human cell, stretched it end to end, it would be about 3 billion bases long and 2 or 3 meters in length. 3 billion bases means that our 4 letters, A, adenine, C, cytosine, G, guanine, and T, thymine, would appear 3 billion times. And that's only one cell. If you could take all the DNA from all of the 100 trillion cells of one human baby and uncoil it and stretch it in a line, that line would be long enough to make 15 round trips from the Sun to Pluto, the farthest planet in the solar system. How's that for efficient packaging? The coded instructions that actually govern the cell's activity come on discrete small sections of the DNA. These sections are called genes. Each gene is itself a sequence of between a few thousand and a few 100,000 or more bases. This sequence of bases on a gene is the code that tells the cell how to manufacture a protein, say a protein like insulin or hemoglobin or one of the digestive juices in your stomach, or one of the combination of proteins that make for the color of your eyes, your hair, your skin, or one of the combination of proteins that make for your height, your stature, your athletic or musical or mathematical ability. Proteins, in other words, are the stuff of which cells and tissues and whole human beings are built and operated. And since genes make proteins, you can see why an understanding of the genetic code is central to an understanding of life itself, all life. Genes, surprisingly, make up only about 3% of the DNA. The rest is sometimes called junk DNA. But this is probably not an accurate term, because molecular biologists and geneticists are already finding it does play important parts in turning genes on and off, and probably in many other as yet unknown functions of the cell. So, how do scientists today decipher that living code? Well, let's take a look.
The basic idea is, so the way DNA sequencing works is there's enzymatic chemistry that allows you to take a piece of DNA and create a set of fragments from the piece of DNA that all have a common start point but terminate in a base-specific manner. So there's these 4 bases, A, C, G, T.
The chemistry of how to break up a DNA molecule into workable fragments was worked out over the last 3 decades using test tubes, centrifuges, special chemical reagents called restriction enzymes, and most important of all, a recent invention, the PCR machine. PCR stands for polymerase chain reaction. Here's what one looks like. They are widely available today for a few $1,000s each. What they do is take one small segment of DNA and, using a heating and cooling routine, they multiply that segment, producing in a few hours millions of exact copies. Millions of DNA clones, in other words. Well, today, much of this molecular biology can be done in automated laboratories, like this one, custom built by Dr. Smith and his colleagues at the University of Wisconsin. Dr. Dave Rank explains.
This all right here is a giant robotic workstation then that can perform all of the molecular biology tasks of shotgun DNA sequencing, from isolation of DNA through the sequencing of the DNA and purification of the sequencing reactions.
In other words, you put DNA samples obtained from a human cell in at one end, and in a few hours you have cut up the DNA into small pieces and then purified and multiplied these fragments. You take these purified fragments to another room where you can separate them from one another by a technique called electrophoresis, and then the actual sequence of bases on each DNA fragment can be detected by a moving laser beam. Dr. Mike Westphal describes how this works.
The DNA goes onto the top portion of the gel, and what the gel is, it's just a kind of a gooey substance between 2 plates of glass, and the substance has a bunch of pores which the DNA passes through. And we load the DNA in the top, and there's what we call a loading comb, which just has a bunch of teeth, and the samples go in between the teeth. And so what happens then is once we load the DNA, we apply a large electric field, and it causes the DNA to migrate through the gel. And so they begin at the top and they migrate towards the bottom. And what this is the detector which is scanning across the gel, and it detects the DNA as it passes through the area in which it scans.
There are multiple lanes in the gel. A different fragment of DNA runs down each lane. Each of the 4 bases has a different dye attached to it. The detector has a laser probe which moves across each lane and identifies each of the 4 bases, A, C, G, and T, as they pass through the bottom of the machine. And it sends this data to a computer nearby.
Basically, after we get done, the data is done collecting on the DNA sequencer we saw down in that room. All that data is stored into a single file. We transfer that file over here, and we reconstruct it with our program, which goes by the name of Gel Imager, which basically forms the image that the scanning detector head saw. And so as you can see, you see all the individual lanes, and those were the separate DNA samples which were loaded onto the gel using the shark's tooth comb. What happens is that sample separates out as it goes down through the gel, exposing its individual bases. And that's what you see. So each of these compose an individual base of a given strand of DNA.
After a special computer program massages this data, you can see it here as many lanes of color blips on the computer screen. Well, you can convert these confusing lanes of data to individual graphs along a horizontal line. Each blip and each blip color there stands for an individual base, A, C, G, or T, sequenced along a fragment of DNA, a fragment of a human gene. This would be one very small segment of a human gene?
Well, that's right. I mean, even this is very small is correct. Well, the sequencing process, as you take your great big long chunk that you're interested in, you keep going through different stages of breaking it into smaller and smaller pieces, and hopefully kind of keeping some order to those pieces. From a great big chunk, we've broken it down now into chunks which are about 40,000 bases long. And from that, we've broken it down into now smaller fragments, which are like 1,000 to 1.5 kb in length. And then you can start tacking those together, and before it's all said and done, you're finally back to your great big piece that you started with.
That great big piece is one human gene, decoded, put now in the computer dictionary as one entry. This automated laboratory at the University of Wisconsin was one of the pioneer labs funded by the Department of Energy and the National Institutes of Health, partners in the Federal Genome Project that began in 1990. At the beginning of the project, the cost to discover just one base in the 3 billion base human DNA sequence was about $10. Building on this technical foundation, the automation technologies and techniques improved dramatically until by the end of the project in 2003, the cost to sequence a single base had decreased 100-fold to less than 10 cents a base. At roughly the same time, Dr. Smith and his colleagues around the world were working under the federally financed Human Genome Project. Another privately funded company, Celera Genomics in Maryland, was also pursuing the same goal using a different automated strategy called shotgun sequencing. Well, both strategies worked, and both achieved their final goal at almost exactly the same time in 2003. By then, both also had an error rate of only one base in every 100,000 base pairs. That's 10 times more accurate than they had originally predicted. The big surprise, however, was that, contrary to almost all expectations, both groups found many, many fewer human genes than just about all geneticists had predicted. It seems clear now that human beings are constructed with a blueprint of only 20,000 to 25,000 working genes, rather than the 100,000 that had been predicted just a few years ago. Well, up until now, we have shown only the basic research. What good will it do?
If you want to understand biology at a really comprehensive level or systematic level, you can kind of pretty quickly imagine that you'd want, at a minimum, to have the information about, what is the biology made of, right? It's hard to analyze something if you don't know what you're analyzing. And basically the sequence of all the genes that encodes the structure of all the proteins is sort of like the minimal fundamental building block that you can use. And although there's a lot of resistance to this concept when the Genome Project started, that resistance has pretty much gone away, in part because the power and utility of this information has become so obvious that nobody can overlook it. So for instance, the yeast genome is now completely sequenced. It's about 10 million bases. And yeast biologists all over the world are all using the yeast genome information all the time. And even yeast, which you might say is relatively simple, 10 million base genome instead of 3 billion, and a unicellular organism instead of a complex organism like us, even there we know remarkably little about, even with all this sequence still, about what really makes this thing tick. How does it divide? How does it control its growth? How does it respond to its environment? How does it shuttle and move and control its gene expression? All of those questions are big, hard questions, which work's going to go on for a long time. And what you do when you produce all this sequence is you give people a very powerful sort of infrastructure tool to help them go after those hard questions.
Scientists now are also rapidly sequencing the genomes of many other species. Besides yeast, the genomic sequences of bacteria, roundworms, mustard plants, fruit flies, mice, monkeys, chimpanzees, as well as many other species of plants and animals have already been sequenced. Many gene sequences are the same in species as different as yeast and human beings, showing once again the basic unity of all living things on this earth. As more and more genomes become sequenced and available to researchers, we will have a more and more complete understanding of the evolution of all plant and animal species on Earth, including our own species, Homo sapiens. And by comparing DNA sequences in humans from around the world, we can already begin to trace a clearer line of evolutionary change and development from our ancestors in Africa to the many ethnic, racial, and even down to the individuals in the world today. So far, we have emphasized the likeness of DNA in all species of living things and talked of human DNA as though it was the same in all humans. Well, it is over 99.9% the same in all humans, but that is not exactly the same. In fact, except for identical twins, each human being has a unique genome. Remember, human chromosomes come in pairs. One chromosome from your mother and one from your father. That means the genes come in pairs too. One set from your mother, one set from your father. In order to make the protein hemoglobin, for instance, you have not one but two genes. If both these genes are exactly the same normal code, there's no problem. You have normal hemoglobin and normal red blood cells. However, sometimes a gene for making this protein can have a mistake in the base code. What is called a mutation happened sometime in the past. That is, the normal gene changed. Maybe just one base in the long sequence got knocked out of order somehow. It's called a SNP. This mutated gene, this SNP, gets passed on to children. In the case of the hemoglobin gene, an unfortunately fairly common mutation can lead to a serious hereditary disease called sickle cell anemia. In sickle cell anemia, the red blood cells take on a sickle shape and cannot as easily carry oxygen to body cells. Now, if one of the hemoglobin gene pair is normal and the other member of the pair is defective, the person will not have the disease. However, this person will be a carrier of the mutated gene. When he or she has a child, the child has a 50-50 chance of getting this mutated gene. Well, if both parents are carriers and each passes on the mutated gene, in the child both hemoglobin genes in the pair would be defective, and the child would have the disease. We think of mutated genes as bad genes, and they usually are. In the long run of evolution, however, human beings are the result of mutated genes that were good. In fact, every one of our 20,000 to 25,000 genes is a successful mutant gene, created and then passed on over 4 billion years by the evolutionary process. And in fact, it is these mutant genes that make us all delightfully different from each other. It is mutant genes, in other words, that have led to the incredible diversity of living things on this living earth. In the short run, though, mutant genes are often a serious problem in the human species. Diseases like sickle cell anemia, cystic fibrosis, Tay-Sachs, Down syndrome, and Huntington disease are all caused by a mutation in a single gene. Now, here is one place where the Human Genome Project may be of great help in the future. Dr. Smith explains just one example of benefits already realized from basic research in the human genome.
There's some good examples of that, actually. Another company I work with called Visible Genetics has been doing sequencing of the retinoblastoma gene. So it's a gene that's involved in generation of cancers of the eye. And it turns out that if you're in a family that has that gene, then and if you don't do any genetic testing, then you don't know which of your kids have those genes and which don't. And it turns out, since you don't know, what they end up having to do is they have to do these examinations under general anesthesia of the eye, which are pretty expensive tests, and they have to do them like every 6 months on young children to detect if there's going to be an early occurrence of this eye cancer, so they can then try to ablate it by laser surgery at an early stage. So it's a very expensive test, and it's very stressful for the children who are exposed to it, and it's stressful for the whole family, and everybody's sort of sitting on a bomb and they don't know when it's going to go off. So Visible Genetics developed this test that allows them to go in and rapidly sequence those genes from the affected members in the family. Once they do that, they can find out what the mutation is in the gene that's causing the problem, and that allows them to then go and very quickly and easily test the children and find out which children have the bad gene and which don't. The ones that don't, right away are free and clear. Okay, they're out of it. No test, no general anesthesia, no anxiety. And so that's a huge benefit right away. And then the ones that do, you can also start building up a database to look at what the prognosis is based on different types of mutations, and also try to tailor treatments that are specific for those mutations. So in general, that's just one interesting example of the social payoff for this. And the social payoff is really going towards targeted treatment for people based on their genetic type, their genotype.
As in all other fields of science and technology, when genuine knowledge increases, there are benefits and there are risks. Automobiles in the 20th century changed the world by making it easier for humans to move around. Few people would want to give up the benefits. On the other hand, more people have been maimed and killed in automobile crashes than in all the wars of the 20th century. So too, the Human Genome Project will dramatically increase our ability to diagnose, to cure, and prevent many diseases. Some critics worry that it will also give us more information than we want to know. Let's say a doctor finds in a routine physical that would include a genetic scan. He finds out that you carry a gene that predicts with high probability you getting breast cancer or prostate cancer or heart attack at an early age. You may or you may not want to know this, but you almost certainly do not want your insurance company to know it. In the future, will insurance companies require this kind of genetic testing as a prerequisite for giving you insurance? Well, legislatures around the country are already considering laws to prevent this from happening. Genetic studies have already shown that at least 5,000 or 6,000 human genes out of the 20,000 to 25,000 total do have mutations that can cause health problems. Some minor, others very serious. Example of the latter would be cystic fibrosis, Huntington's disease, diabetes, muscular dystrophy, and perhaps Alzheimer's disease and some forms of cancer. As with sickle cell anemia, many people are carriers of these genes, but do not actually have the disease because the gene on the opposite chromosome in the pair is normal. Well, knowledge gained in the Genome Project will enable technicians to test and discover whether you carry a mutated gene in your unique genome. What do you do when you find out that you do carry this gene and your spouse also carries it? In that case, there's a 25% chance any children you have will inherit the disease. Now, here's one possibility that has already been used. Using new in vitro fertilization techniques, doctors can genetically screen a number of very early embryos while they were still in a culture dish and find out whether they have the gene pair that produces the disease. then only embryos that do not carry the mutated gene pair will be implanted in the mother's uterus. Of course, using the same technique some critics have pointed out, couples could in the more distant future not only prevent serious diseases from being passed on, they could also choose among desirable genes for their offspring. While there is no single gene that leads to good looks, or intelligence, or athletic ability, or musical genius, there is a strong genetic component to these traits. But when the complete human genome is worked out, we will probably know more about which genes are involved in these complex human traits. Armed with this knowledge, you might be able to screen embryos for genes that would lead to children with not only no genetic defects, but for children with desired sex, height, body build, athletic talent, musical talent, mathematical talent, and so forth and so forth. Some have even envisioned the day will come when prospective parents could sit before a computer screen and choose among many alternatives what traits they want their next child to have. It's not inconceivable that cut and paste techniques already developed in other plant and animal genetics could then be used to insert desirable genes or remove undesirable ones. There is even the possibility of adding an entire chromosome with many novel genes to the embryo, genes never before a part of the human genome. Other animals, for instance, have genes for seeing infrared or ultraviolet light. You might be able to insert a gene for such a capability into a human embryo. Or you could insert a gene to enable the offspring to detect odors as well as a dog or a cat, or to navigate by sonar like a bat, or generate electricity like an eel, or navigate magnetically like a bird, or, well, Leo M. Silver, a professor of molecular biology at Princeton University, has gone so far as to predict that such genetic screening and reconstruction is not only possible, but inevitable. He thinks that by the 22nd or 23rd century, this may well be the norm. That is, for educated, well-to-do parents who can afford the expense. What about parents who either cannot afford the expense or decide on religious or moral grounds they do not want to interfere with nature in these dramatic new ways? Well, the long-term effect of this split may be surprising, says Silver. Controlling human genetic continuity for the first time in history may lead to a truly revolutionary change in a few centuries, a blink of the eyelash when it comes to evolution. The well-to-do, scientifically oriented humans, say Silver, may well make themselves into a different subspecies from natural humans. And in fact, human-controlled evolution may accelerate to the point where there will be at least two varieties of Homo sapiens, so distinct in their genomes that they will be actually unable to mate and produce fertile offspring. Silver calls his book Remaking Eden to call attention to the revolutionary events that may accompany that step into the genetic unknown. Whatever actually happens in the 21st century, it seems certain that the words of Neil Armstrong, when he first landed on the moon, will apply even more aptly to the Human Genome Project. A small step for man, a giant step for mankind.